Hello everyone, so today we'll be looking at cellular respiration, part 1 of 3. So we'll be looking at harvesting energy from carbohydrates and other fuel molecules, and we'll be looking at glucose oxidation and energy released. So we'll see this figure quite a bit, but we finally got to 3. So essential features of cells are that they can harness energy from the environment. So the structure of macromolecules allows energy to be stored that can be released in metabolic pathways using special proteins. So we have two metabolic pathways we can see here. We have our catabolic and our anabolic. So catabolic metabolic pathways harvest energy of carbohydrate as ATP, whereas anabolic metabolic pathways capture light in this example and are stored as chemical energy. The pathway used depends on the presence or absence of O2. So with oxygen we have aerobic conditions, and without oxygen or anaerobic we have anaerobic conditions. So chemical energy stored in carbohydrate bonds is extracted and converted into usable energy through ATP. So in most cases, the sun, through photosynthesis, our plants use photosynthesis to create glucose. Then animals either eat these plants, and then we eat those animals, or we eat these plants to get our glucose. Then glucose will go through glycolysis. This creates pyruvate, and this can enter two pathways, aerobic or anaerobic. So in anaerobic conditions, we have fermentation, where we have incomplete oxidation, and our waste products are organic compounds and CO2, and our net energy is 2 ATP. In cellular respiration, we have complete oxidation, we have the waste products of H2O and CO2. So again, this is that waste product we need to breathe out with respiration. We also need oxygen for cellular respiration, as we can see. And the net ATP is 32, so you can see it's a much higher net gain than without oxygen. So how does glucose oxidation release energy? So here's the overall reaction of the oxidation of glucose to CO2. So we have our glucose here, 6 oxygens, and then again, here's our output, our waste products, and then we have energy. So when glucose is oxidized to CO2, it is through combustion, which occurs in one step, where energy is released as heat or light, or metabolism, which occurs in many steps. So this is a series of metabolic pathways catalyzed by enzymes. So enzymes are also known as catalysts. So we have energy transfer through ATP, through phosphorylation. So this is when we have our ATP to ADP, with the phosphate being released. And we also have energy transfer involving electrons using redox reactions, which we'll get to shortly. So energy transfer using ATP. So as we mentioned, there's a phosphorylation. So there's addition of the phosphate molecule, or the PO43 or PI, or inorganic phosphate. This is an endergonic reaction, where energy is consumed. So this is to create ATP. So an ADP, which is adenine diphosphate, an inorganic phosphate, plus 3 energy equals ATP. So during dephosphorylation, so these are types of reactions, just to clarify, we have the release of the phosphate molecule through hydrolysis. This is an exergonic, so think of exergonic as exit, energy is released, endergonic, energy enters, is consumed. So we can see here I have ATP, and with the addition of water, there's the release of that free energy when we break the phosphate groups. Now we have adenosine diphosphate and 1 inorganic phosphate. And again, these are usually recycled. So we can also have energy transfer using the redox reactions. So some key terms here are reduction and oxidation. So reduction. Reduction is the gain of electrons. So when you think of reductions, think of it in math. It's easier to think of it in math as in negativity. So we know that electrons are negatively charged. So if we add an electrically negative charged molecule or electron to something, its charge will be reduced, meaning it becomes more negative. So we can have the gain of electrons, again, are negative, or hydrogen atoms, or a proton, which is an H proton electron. So this is usually denoted as hydrogen ion. This is endergonic, again, energy is consumed when we gain. So in oxidation, which is the loss of electrons, So if you think about it again, losing electrons are going away. This cell will have a lower negative charge. So it's the loss of electrons or hydrogen atoms, including the proton electron, as we said. And this is exergonic. Energy is released. So here we have a reduced compound A. This is the reducing agent. And then we have the oxidized compound A. So here in reduction, we lose the electron. So here there's 2 electrons. Here we can see. So now oxidized compound B, which is the oxidizing agent, gains electrons. So we can see here. So these are working together. So when we that's why it's called redox. In these reactions, something is getting reduced and something's getting oxidized. So if something gets reduced, something else must become oxidized, essentially. So this energy is liberated by oxidation of the reducing agent A, which we can see here is considered the electron donor. So if we follow the blue, the electrons are being donated, the reducing agent, to compound B. Here we can see it's the electron donor. These are captured in the reduction of the oxidizing agent B, which is the electron acceptor. So again, here's our overall reaction. So we can see here carbon atoms and glucose are oxidized. And here we see carbon atoms share electrons equally. So here's our carbon-carbon. So this carbon valence is 0. The carbon atom has partially lost electrons and is oxidized. So remember, oxidizing is the loss of electrons. And because the oxygen atom is more electronegative than the carbon atom, we can see here the dark shading represents increased electron density. Now the carbon atom's valence is positive 4. Carbon becomes oxidized. So here we have an oxygen atom and water is reduced. So here's our oxygen, here's our water. The oxygen atom shares electrons equally. Valence is 0, as we see. Then the oxygen atom has partially gained electrons, so it is reduced. So remember, reduced is gaining electrons because the oxygen atom is more electronegative than the hydrogen atoms. Then a -2 valence. Oxygen becomes reduced. So in glucose oxidation, glucose is the reducing agent. It is the electron donor. And oxygen is the oxidizing agent and is the electron acceptor. So you'll sometimes hear that oxygen is the final electron acceptor in the electron transport chain. But we'll get to that later. But that's important. So energy is transferred in redox reactions involving intermediate energy carriers. So example of these are NAD plus and FAD, which we'll talk about in the Krebs cycle and in glycolysis. So the energy of reducing agent is associated with the reduced product. So here's an example in a glucose. So here we have our glucose, which we said here is the reducing agent. And we have our byproduct of CO2. And here's the intermediate energy carriers, NAD plus, as well as NADH. And here we have our H2O. See here we have oxidation and reduction. So the structure of NAD. So NAD stands for nicotinamide adenosine dinucleotide. So NAD is a coenzyme and is an intermediate energy carrier. So here's the structure of NAD, of the entire thing, and then here's our nicotinamide. And then the other ones include our adenine dinucleotide. So here, like we said, here's our nucleotides and our adenine. So remember, adenine is one of our base paired nucleotides. And then here we can also see our phosphates. So again, it's a coenzyme, and it has 2 parts, the nicotinamide nucleotide and the adenine nucleotide. So here's the adenine nucleotide in purple, and then a nicotinamide nucleotide in green. So there are 2 forms of NAD. There's the oxidized form, so that's important. Oxidized form is NAD+. So remember, if it's positive, you can kind of think of, again, oxidized is loss of electrons. And the reduced form is NADH plus hydrogen ion. So remember that we said the hydrogen ion can also be in the same, also be an electron or a hydrogen ion. So this is considered free. So here's an example. So this contains a positive charge while this contains a neutral charge. So you can see the difference here. We've got that extra hydrogen ion, but here we don't have it. So in this redox reaction, we can see that we're having the reduction here and the oxidation here. 2 hydrogen ions, or 2 electrons and plus 2 hydrogen ions. We can see here, plus 2 are being moved. This acquires 1 electron and 1 hydrogen ion, while 1 free. So the function of NAD in this scenario is the energy carrier. So essentially, the energy of NADH plus H is greater than the energy of NAD plus. Oops, sorry. So this is the end of our short part 1 lecture. So here's some suggested learning for metabolic pathways, but we'll also answer some of these as we progress. So who uses metabolic pathways? Why? Where are they taking place? We'll talk about this later, but they're different: glycolysis, electron transport chain, and the Krebs cycle. What are the reactants and products? What is happening with the carbon compounds? That's quite important, especially in the Krebs cycle. We tend to follow the carbons. What is happening to free energy levels? So how much energy is being used or produced? What is happening to ATP? How many we used or how many did we make? What's happening to the carriers? So a lot of these carriers will be used in certain reactions, but then become recycled back up the chain or used later in the chain. And then how is the pathway regulated? So a lot of these are regulated in multiple different ways. It can be upregulated or downregulated. Anyways, I hope you learned something. See you in the next one. Take care.